Now that we've finished learning about how the induced fit model of an active site is able to bring the substrate into the active site, it's time to actually look at what the active site does during the process of catalysis. And in order to do that, we need to look and consider the function of enzymes as biological catalysts from the perspective of what they do to the energy in a chemical reaction. So for those without a strong chemistry background, we're going to be looking at something that we call activation energy. Now, the word activation, very simply, is a complicated way to say the word to start. So activation energy is the amount of energy that you need to put into a chemical reaction, specifically the reactants of that chemical reaction, in order to get them to basically spontaneously react with each other and complete the reaction by themselves. For example, if we take a look at the reaction being catalyzed here, we have a disaccharide molecule, and this is most likely sucrose, a combination of a glucose monomer and a fructose monomer. There we go. Now we can see that sucrose is attracted into the active site of the enzyme, and this we see is definitely induced fit going on, because once it enters the active site, we see that the active site changes shape. Now, technically, we can see that this is most likely a hydrolysis reaction because the enzyme is adding water, resulting in the bond between glucose and fructose being broken, and we can see that the product molecules are individual monosaccharides. Now, technically, this hydrolysis reaction is possible without an enzyme. All that you would need to do is drop some sucrose into a large enough supply of water and most likely increase the temperature, which would increase the possibility of water colliding with the fructose molecule and causing a hydrolysis of this bond here. The problem is, without the enzyme, you're relying a lot on random chance that one water molecule collides exactly in the right orientation to break this bond and has enough energy, what we call the activation energy, to actually result in the bond here changing structures. So all that the enzyme does is it makes this process of water colliding with the sucrose molecule easier and more likely to occur. The first and most obvious way that an enzyme does this is that it uses its active site in order to attract the substrate and basically hold the substrate in place so that the water has a much greater chance of having a successful collision with the exact part of the sucrose molecule that we want to break. Now, this we've already talked about in the induced fit lesson, but we can do a little bit of a review. So because a sucrose molecule is a disaccharide, like all carbohydrates, we would expect that because of all the OH groups that you find on the outside, that this molecule is very, very polar, meaning that our enzyme will most likely have complementary poles to form hydrogen bonds with the sucrose molecule in order to get the sucrose molecule and only the sucrose molecule to enter the active site where it can be held in place more easily for the water molecule to collide with and hydrolyze this bond. The second most important consideration about the enzyme to make is the effect of the conformational change. Remember that the conformational change is the actual shifting of the enzyme structure that happens when the intermolecular forces of the active site change after the substrate has entered it. And we can see that the walls or the outside of the active site are kind of putting compression forces on the sucrose molecule here, resulting in this bond being put under strain, and we'll talk about this in a second. Now, depending on what the reaction being catalyzed is, the conformational change can have a number of possible effects. One possibility is that it brings multiple substrates closer together, and this would be very useful because if this enzyme were catalyzing a dehydration synthesis reaction, you would want the two molecules being joined together to be closer together so that it's easier to form the bond between them. One other possibility, if we were doing a hydrolysis reaction like this one, is that the enzyme uses its active site to pull a single substrate farther apart, thereby, again, putting strain on the bond that we want to break. And because it is the conformational change that is putting this bond under strain and making it want to break, we would naturally call this conformational strain. And it is this conformational strain that makes what we have here, what we call, remember, the enzyme substrate complex. This conformational strain is what makes this enzyme substrate complex inherently unstable. In other words, the enzyme makes it much more easy for the substrate to undergo the reaction that it wants. So as mentioned previously, if the enzyme brings multiple substrates together, this makes it more likely that those substrates will form a new bond or even new bonds, depending on the complexity of the molecule. Whereas if we take a single substrate like sucrose and put the substrate's linking bond under strain, this makes it easier to break a bond or possibly, again, multiple bonds within the enzyme itself. Now, because the enzyme's entire reaction is different than how this reaction would happen outside of an enzyme, this means that the enzyme has actually altered the mechanism, which is the specific movement of electrons between the different reactant molecules in order to change the bonding structure. So depending on what an enzyme actually does in its mechanism, this may take a greater number of steps than the original process without the enzyme, or it could make the process more streamlined and happen in fewer steps. This is inherently impossible to predict because it depends on the enzyme and the starting reaction that it is trying to catalyze. But the point is, the conformational strain created by the active site either makes it easier to form a new bond, or in the case of this hydrolysis reaction, to break a bond once the water is added in. I should rather draw the water being added here, and now we can see that the bonds are broken, and because these individual monosaccharides no longer fit into the active site that has undergone conformational change, they will simply be pushed out of the enzyme, allowing it to be reused. Now, if we represent the change in activation energy on a potential energy curve, for those that don't know how to read this, we always put the time that it takes to do the reaction on the x-axis, and we put the potential energy, that being the amount of energy that shows how capable a substance is of reacting. And if our reactants are here, including our substrate, and if these represent the products, the molecules produced after catalysis, this distance here represents the activation energy, which is what we call E sub A. This is the energy that needs to be inputted into a reaction in order to get the energy to, or in order to get the reaction rather, to start. And what we have up here is what we call the activated complex. This is the structure that forms when all reactants successfully collide with each other, allowing the reaction to begin. Now, like inorganic counterparts, catalysts that are not protein or RNA based, enzymes, or the job of enzymes, is to decrease the activation energy, and we can see that here. So the highest point that our potential energy requirement is now is here, and this is equivalent to our enzyme substrate complex, because remember, the job of the enzyme is to grab hold of our substrate, put it in the active site, and hold it in a specific position that makes it more likely to react, therefore decreasing the amount of energy we need to add in order to get this chemical reaction to start. Now, we can see that the graph looks completely different in the catalyzed version of this reaction because, remember, the enzyme also changes the number of different steps that are required because it basically alters the entire mechanism of how a catalyzed reaction happens compared to its uncatalyzed counterpart. In the next video, we're going to look at different environmental factors that can both drastically speed up or drastically impede the ability of enzymes to perform their given function as biological catalysts.